feel it moving, but I was particularly interested in extracting 3 different sort of sections of DNA from Richard. So the first of these was mitochondrial DNA. Now, mitochondrial DNA is interesting for this project for 2 reasons. First of all, it's in lots and lots of copies in each of our cells. So if you're going to find any DNA left in skeletal remains, it tends to be mitochondrial DNA. The second reason why it's really useful for this case is it just has a really simple pattern of inheritance. It just goes down through the female line. So, mum passes it down to all of her children, boys and girls, but only daughters pass it on. So it means that if I can find a female line relative of Richard, and it can be a boy or a girl, as long as it's all female line, all the way through the rest of that line, I can use them to act as a comparator. I can look at the living individual's mitochondrial DNA and see if it matches those of the remains. The second bit of DNA I was interested in was the Y chromosome. So the Y chromosome also, very simple pattern of inheritance. It has on it the gene for maleness, so it only goes down through the male line. So all men who were related through the male line should have an identical or near identical Y chromosome type. And the third bits of DNA that I was interested in are bits of DNA that we know can be used to predict quite accurately somebody's hair and eye color. The reason why that's interesting is because there's no contemporary portraits of Richard. All of the portraits of Richard post-date his death by about 25 to 30 years onwards. So what I was interested in doing is looking at what the DNA tells us about what his hair and eye color looks like, and then comparing that against the portraits that we've got and seeing which is the closest match.